With Doubtnut, get instant video solutions to all your maths, physics, chemistry, and biology doubts. Just click the image of the question, crop the question, and get instant video solution. Download Doubtnut app today.
So the question is the concept that explains how the large quantity of substrate is acted upon by a small concentration enzyme is, and the options are A. Coenzyme theory, B. Enzyme collision theory, C. Induced fit theory, and D. Lock and key theory. So here we have to say the theory. So in the biochemistry part, specifically in the enzyme kinetics. Okay, so in the enzyme kinetics, we all know that an enzyme is being, it acts as a, so it is a catalyst, okay, and the two partners, okay, which are involved in this reaction, enzyme is reaction is a substrate will be there, and of course the enzyme will be there. So this is the basic rule. Okay, so substrate binds to the enzyme, and then eventually the enzyme product complex will be formed, and then product will be released from the enzyme. So this is the overall gist of that. So now here we have to see the specific theory, or we can say specific concept that explains how the large quantity of substrate is acted upon by a small concentration enzyme, so that versatility is maintained, so you can say. Okay, so now let us go option by option. So option A is coenzyme theory. So now coenzyme, there is no such coenzyme theory. So coenzyme is basically, it is a part of enzyme. Okay, so it assists, you can say, it assists the enzymes for the completion of reaction. Okay, so that is the contribution of coenzymes. Okay, along with, so you can say cofactors. Okay, cofactors are the major chunk. So cofactors is equals to coenzyme plus an inorganic metal ion is there. Okay, so inorganic ion like zinc and all. So that is the thing. Now let us go option B. Option B is enzyme collision theory. So this there's no enzyme collision theory. Generally, the collision theory states that the rate of reaction, okay, rate is proportional to number of collisions. So more the number of collisions, more the rate, higher the rate of reaction will be. Fine. And then the induced fit theory states that the when a substrate, okay, so if I draw like this, okay, this is the enzyme and the substrate binds to this, okay, then this is being modified, and so in, say, in this, the active side, this is the active side, substrate binds to this, then the enzyme structure can be modified, and eventually it can lead to either activation. Okay, so that means more substrate can bind to this enzyme, that is activation, or inhibition, that means the another substrate which can possibly bind to another some other site is actually now inhibited. So both these things can happen. That is actually the induced fit theory. Okay, so all these does does not relate to this versatility of the enzyme, which is the question, matter of the question. But lock and key theory, now if you see, that is actually the theory which explains properly how the large quantity of substrate is acted upon by a small concentration enzyme. Okay, so why? Because lock and key theory states that first of all E is there, that is enzyme. Okay, then eventually the substrate comes, then enzyme substrate complex happens. This is the lock and theory. This is a part of that only. This the enzyme substrate comes, there's a lock kind of thing happen. Okay, lock and key, so properly, and then what happens, this is converted to enzyme product complex. Okay, and then this lock is being unlocked, and then product plus enzyme. So this is lock and key hypothesis, and this explains well the versatility of this enzyme. So the correct option to this question is option D, lock and key theory.
For class 6 to 12, IIT JEE and NEET level. Trusted by more than 5 crore students. Download Doubtnut app today.